Hi everyone, I'm Dr. Roger Sweets, and I have the honor of presenting the information in Chapter 14 of Campbell's Biology to all of you folks. Now, Dr. Stemke has spent some time talking to you about mitosis and meiosis, what's going on down there in that cellular level. And now we're going to explore a little bit more about what all that means for genetics and the appearance of organisms across the spectrum, as well as their evolution. We're going to do that by exploring what we call Mendelian genetics. And I'm going to start off by emphasizing for you a little bit within your text, which I hope you have actually got. It's really useful to look at these concepts that they list in the various chapters. And I'm going to go ahead and organize my lecture in that way. So, especially at the back of the chapters, they give lovely little summaries of all these concepts and some of the ideas behind that. So make sure you take a look at that, as well as browse through some of the chapters, perhaps as you watch these PowerPoints and so forth. So with that, I'm going to go ahead and share my PowerPoint. We all wait with bated breath. Okay, so I'm starting off with concept 14.1. It is chapter 14, which Mendel uses a scientific approach to identify two laws of inheritance. Well, that all sounds very dry, but in fact, it was an incredibly important advance as far as the science of evolution and biology came to be. Because at the end of this process, Mendel understood, even though he wasn't recognized in his own lifetime, that the inheritance of traits is particulate and not blended. Now, to explain why that's important, it's going to take me just a couple of minutes. I want to sort of go back to some other kinds of ideas. So I'm going to start off by looking at this great picture of the garden this is the remains of the garden. The communists tore it down and had to be refound. He had a greenhouse and he had a garden. This is in Burko, I believe, now in the Czech Republic. It was in Austria at the time. And of course, as you know, he was a monk, and he was at the Abbey of St. Thomas, I believe. And he came there. He was there originally when he was quite young, when he was 21 years old. And after he was there for a while, he went out to the University of Vienna and he was trained a little bit further in some of the things he was already interested in. He was quite a scientist. He was not just a guy that played around with peas. He did all kinds of interesting studies in astronomy and geology and all kinds of things. This is sort of the remains of his garden. And in a way, this was a revolution that happened, a quiet revolution that happened when nobody actually knew about it. Now, one of the things that's at the heart of this is something that Dr. Stemke probably finished with, which is the sources of genetic variation in eukaryotes. So I'll come back to what variation really means. This was concept 13.4. And there are really three main sources of genetic variation in terms of eukaryotes. There are some others in prokaryotes. We won't get into those right now. And those are crossing over, which happens in prophase 1 of meiosis. Hopefully you remember what that is. I won't describe it again. It turns out to be vitally important. In fact, in some ways, the most important source of variation within our common genetics. The second, the independent assortment of chromosomes as they line up along that metaphase plate. Remember, Dr. Stemke probably described that. And in fact, that description was something that Mendel himself discovered, even though he didn't know anything about genes or chromosomes or things of that nature. That independent assortment was the most important part of that in metaphase 1 of meiosis. And that's what he called the law of segregation. And in our terminology, it's a law of segregation of alleles that occurs when homologous alleles separate. And then, of course, when mating actually occurs, there's a random fusion of whatever gametes are involved in that process during fertilization. Now, to understand a little bit about why this is important, and I won't go through all these, although this is a slide you might want to pause on for a second as you watch this and study it rather carefully. Because the modern synthesis, of course, is the main paradigm of biology. It's the melding of genetics and Darwinian evolution. And here we see some of the most important things that are part of the modern synthesis. As a result of genetic recombination, individuals within populations are variable for many traits. We can go ahead and pause right there. Although, if you look down the line, a lot of these other things actually involve Mendel's findings as well. If, in fact, when organisms were to mate and there was a blending as opposed to a particulate passing on of traits of some kind, then everything would end up being the same. In fact, variability would disappear. Variability is still an interesting situation within evolution and biology. But at this point, at this time, it was mostly believed that traits, for instance, a purple flower and a white flower, when they were crossed, that they would come up with something in between, what we call blending inheritance. And in fact, that was not a silly thing to believe. There were many instances in which that actually appeared to be the case. We'll get to that a little bit later. This particular lecture is going to involve just concept 14.1 and 14.2. And later on, when we get to concept 14.3, we'll see how it is that, in fact, sometimes you will see something that seems like blending. But the important thing is that even when blending did occur, there was still this particulate basis to how it happened. And that's what Mendel actually discovered, and that was a great advance. Darwin himself said, although he didn't understand exactly how it happened, that for his evolutionary theories to be true, that in fact the inheritance of traits had to be particulate and not blended. Okay, so we'll go from there to this blending inheritance problem I just described. And we'll talk about Mendel. Now, Darwin wrote The Origin of Species at the same time that Mendel was performing his experiments. And after just a few years after the time that Mendel that Darwin actually wrote Origin of Species, which I think was 1859, it was about 1864, 1865 that Mendel presented his papers on what we're about to describe. Okay, so Gregor Mendel, from his own experience, had begun to think that perhaps particulate inheritance was something that was going on in at least some organisms on the planet. And he'd been sort of interacting with botanists, and the monks at the Abbey of St. Thomas were very involved in planting organisms and following what was going on, and raising their own crops and so forth for their own eating pleasure. And what Gregor Mendel was able to do was to really use a very strong experimental basis, which he had been trained in, which is a little bit different from most, in order to demonstrate his hypotheses. He himself didn't know exactly what he would find, but at least he knew how to go about finding out how to do it. Now, before we get into that, some quick terminology phrases, and certainly this chapter is full of incredibly important terminology. We have, in Mendel's terminology, a character, or sometimes called a factor of inheritance, which is like pea flower color. And then there was a trait within the pea flower color, such as the variance of color, such as purple or white. He could have plants that were true breeding, in which they always produced the same variety. And he would do that because the plants he was dealing with, peas, which was a great choice, by the way, they produce all kinds of flowers and all kinds of seeds. And they would pretty much always self-fertilize. The flowers had both the stamens and the pistils, and so they would actually fertilize themselves. And so if you raise them carefully to make sure they were always, in fact, self-fertilizing, he would raise them generation after generation after generation, and he would get true breeding plants with the various traits that he was going to experiment with. Then, of course, you could hybridize those two breeding varieties and find out what actually happened. We often will refer to, in these various problems we'll give you and things like that, that we have this P generation, the parental generation. We'll have the F1 generation, which is the hybrid offspring of that parental generation, and then an F2 generation when we cross those F1s with each other. An important step, by the way, for Mendel. Okay, so right off the bat, what Mendel did, fairly straightforward. He had these true breeding flowers in which he was doing all kinds of things, as we'll see in a second. He actually, I think, started off with peas and how wrinkled they were, but we're going to use flowers as an example. So he had true breeding purple flowers, true breeding white flowers, and he had to stop them from self-fertilizing because he wanted to hybridize them or crossbreed them. You could do that by clipping out the stamens, that's the male, the parts that produce the pollen. He would clip them out so they couldn't self-fertilize. Then he would take a little brush, he would take the pollen from one flower and put it into the other. In this case, he was breeding purple flowers and white flowers. So when he did that, they produced the seeds, the peas, obviously, and he would plant those and he would see what came up. Now, of course, if it was blended, then you would expect them to be somewhere halfway between a purple and a white flower, like a pale purple. And when you did that, in fact, that first filial generation, that F1, turned out to be all strongly purple. So actually, right off the bat, he knew the information was not blended in his examples. And that had been seen before in other ways, but what Mendel did that was a little bit beyond that was that he went ahead and bred that F1 generation with itself. And what happened, of course, as you know, is that that lost information, the white, which disappeared within that first generation, suddenly reappeared. So we had the P generation of true breeding plants, the F1 generation, the hybrids, all plants have purple flowers. He let those actually go ahead and cross-pollinate with themselves. And then that F2 generation came up, and somehow the factor, which is what the term he used was, for white, had been hidden in the F1 generation, now reappeared in the F2 generation. That's not all, because he had been taught good scientific principles. He counted each one of those flowers that reappeared and kept track of that over many replications of the same experiment. This example here, we see there were 705 purple flowered plants, 224 white flowered plants. Now, that's not exactly a 3-to-1 ratio, but as he experimented more and more with this, it always hewed pretty closely to that 3-to-1 ratio. And that, of course, got him thinking about how that could be. He repeated that experiment with all these different kinds of traits, with true breeding varieties of peas that had yellow seeds or green seeds or wrinkled seeds or round seeds, or all these examples you see here. And as you can see, the ratio pretty much always came out 3-to-1. In fact, I think the round and the wrinkled were one of the first ones that he did, and they came out really, really close to 3-to-1. So we saw this pattern coming out, whether it was flower color or something to do with seeds or something to do with actually the plants themselves being tall or short. He began to understand that something rather interesting was going on. And so we can account for this with 4 findings that Mendel put forth. I'm going to put most of these in our modern terms, which of course he didn't know about. Alternative versions of genes account for variation in inherited characteristics. Here we are with that magic word variation that Darwin required. Now, in this case, alleles are going to be what I called originally traits. Those would be purple and white. Those are 2 different types, versions of a gene, or what we often call a locus, which is like a location on a chromosome. But we'll get to that a little bit later. And that was like the character of flower color. So the trait was the gene of the specific, the trait was a specific color. That would be like an allele. And the gene overall would be some location where the plant was actually coding for that flower color within the DNA. Now, for each character, then, an organism appeared to inherit 2 alleles for that gene. This is what he found out from his 3-to-1 ratio. And it would inherit 1 from each parent. Now, Mendel didn't know anything of genes, but he knew how to count. He got to 3-to-1, and by doing some very simple math, he deduced this idea of particulate inheritance. That, in fact, he had this purple trait and this white trait could be passed on into that F1 generation. And that white was still there because it was a particulate thing, but didn't get blended together, and it could reappear in the F2 generation. And so because he saw this all purple in the F1, if those 2 alleles differ, he decided that one was dominant, really affected the appearance, what we often will call now the phenotype, and the other is going to be recessive, did not affect the appearance. Now, this is not always going to be so, believe me. This simple case we often call simple Mendelian genetics. And Mendel was extraordinarily lucky in his choice of plants and traits because of things, again, I'll talk about when we get to a later part of this series of lectures. But luckily for him and for us to understand this base idea of how genetics works, this particulate inheritance, it came out with this appearance where one was always dominant and one was recessive in some way. In modern terms, we would say that, in fact, what happened, if you think back on what Dr. Stemke had talked about, is that there would be, for instance, an allele for purple flowers and an allele for white flowers. One would come from mom, one would come from dad, or the other way around. they would be combined together in some DNA, and then, of course, it would produce an enzyme. Now, of course, it could be you have two copies of DNA, as you see here. Let me use my mouse real quick. Two copies of DNA, and in every cell. And we would see that this enzyme was actually synthesizing a purple pigment. What was going on with the white flower all that time? It would produce no pigment at all, no enzyme whatsoever. So in fact, they're white because there was no pigment being produced. So in this particular instance, when you had a purple flower and a white flower, you crossed them together. The white allele was still in those F1 generation, but it just simply didn't produce the enzyme. This was producing an enzyme, and it was apparently enough of the enzyme to produce a strong purple color. We'll come back to what that means a little bit later. So this was his first of his great laws, the law of segregation, that these alleles, when inside of an individual organism, would then separate always, even though they would be closely aligned in some way inside of what he didn't even understand at that time, they would become segregated into separate gametes. In other words, every egg and every sperm would only contain one of the two alleles. And so we see now the F1 generation here. So let's start with the purple. Here we see our true breeding, which are going to have a typical sort of typical sign that we'll use, a capital letter for the supposedly dominant trait and a small case letter for the recessive trait. So these all had the large, right, the gametes. So even though an individual flower and the cells within that flower would have two copies of the dominant trait, the gametes would only have the one. In other words, they'd have basically half of all that would be going to a male or female. And the white flowers would have the recessive trait, right? So all of those gametes, because they only had the one kind, they were in fact homozygous, all of their gametes would have one or the other. Now we get to the F1 generation, when obviously each one of the cells would have one of the alleles for the purple and one alleles for the white. And then, of course, those would also separate when the gametes were made. And so we can expect then that half of all the eggs would have the dominant trait, and the other half would have the recessive. And the same would be true of the sperm, pollen in this case, that half of them would have the dominant and half would have the recessive by simple laws of probability. And in fact, when we cross those, we can see what happens. We now have our ova, there's our female right here, and we have half of them with the dominant purple trait, and we have half with the white trait. Here we have in the sperm, or the pollen, half with the purple dominant trait and half with the recessive trait. And then these would come together in some random fashion. I'll come back to how that works mathematically a little bit later. And so now we see within this first of the Punnett squares that I'm going to show you, we see that we have the two large P's for the purple together, just like the parent plant. We have a large P from the male. We have the small recessive white trait from the female together, but of course it still looks purple because that's a heterozygote trait. And we see the opposite way they came together here with the dominant trait, the purple, coming from the female this time, and the recessive trait, the white, coming from the male. And then we have another heterozygote. And then, of course, we have here the chance of having our two gametes being both white, the recessive trait, and that would come out at a 3-to-1 ratio. Now, why would that come out as a 3-to-1 ratio? Let's go ahead and look just a little bit deeper here at these various combinations that we see. First of all, a little bit, I'm sure this is a review for almost all of you. Just a little bit of terminological review here, right? Terminology review. We have the phenotype, which is the outward expression of the genetics. Now, we think of it as appearance frequently, but it can be a lot of different things. It could be behavioral, it could be physiological, but it's the outward expression of whatever that gene is actually doing, typically, of course, producing an enzyme that has some effect. Then we have genotype, which refers to the genetic makeup. We can see that already in this very simple example that we have here. Here's our appearance, purple or white. Here is the genetic makeup associated with that. Same thing in the F1 generation. The appearance is purple, but the genetic makeup now is a different combination, right? Not homozygous. Well, you have identical alleles for each gene, identical alleles within this gene, but heterozygous. We have different alleles for the gene, but only one is being strongly expressed, in this case, the purple. So keep these in mind. This will come back over and over and over again in the next few weeks. This is a different way to look at it because it demonstrates a little bit that we have a phenotype that is 3-to-1, but beneath that in the genotype, the actual source of this, we see that one of the purple phenotypes, or 1/4 in this case, is being produced by a homozygous individuals. We see that about half are being produced by heterozygous individuals, but still purple in the phenotype. And of course, we see that every time we have this homozygous recessive, then we see the white phenotype that goes along with that. So that means the phenotype ratio is 3-to-1, but the genotype ratio is 1-2-to-1. Now, as we go forward into our genetics problems, which is going to happen just in the next few days and hours in class, this is something important to keep in mind, and we'll review this as we go through. In fact, if I go backwards, you can see this right here. We can see that while we have 3-to-1 in the phenotype, we have 1-to-1 in the genotype. Let's get crazy, like Mendel did, because he was a pretty crazy guy, let's face it. And we're going to do something a little bit different. So what we just looked at was what we call a monohybrid cross. The F1 generation were heterozygous for just one character. Okay, but what if we expanded that a little bit? And we produced, for instance, by certain kinds of crossings, we were able to get an F1 generation that was heterozygous for 2 characters. If we were true breeding and we were really controlling things, we could go ahead and take, for instance, a round pea and a wrinkled pea that went along with a purple flower and a white flower. We're going to call that a dihybrid cross, and that's what Mendel did. But before we get crazy, and this is concept 14.2, we're going to get a little bit mathy. And concept 14.2 is that probability laws govern Mendelian inheritance. And this can be important because that gives us the tools to figure out trihybrid crosses, quad hybrid crosses, infinite number of hybrid crosses if we know what's really going on. Okay, so here we have this original example that I gave you. I'm going to go ahead and expand this a little bit in this way. So as we talked about before, just by simple chance, we figure in this heterozygote right here, as it produces ova, we figure that since it has this heterozygous, each cell is heterozygous, each sex cell is heterozygous, as it separates with our independent assortment into separate gametes, about half the time we should get the dominant trait into half of our ova, and half the time we'll get the recessive trait into our ova. And the same thing will happen with the sperm. And now we can look at sort of the math of that and say, okay, so if this is 1/2 of all of the ova, and this is 1/2 of all the ova, and this is 1/2 of all the sperm, and this is 1/2 of all the sperm, maybe we can do some simple math. Punnett square is great, but if we know these math rules, we might be able to go a little bit further. So what we have here is we can say, well, 1/2 times 1/2 equals 1/4. Okay. Hopefully you know enough math to figure that out. You could say 0.5 times 0.5 is 0.25, either way. So 1/2 times 1/2 equals 1/4. We see here that those are for our 2 dominant traits, the big P's. Here we see 1/2 small p from the ova, large p from the sperm come together. 1/2 times 1/2, so 1/4 are formed in that fashion. It's a heterozygote, but in that case, the dominant trait came from the female, and the recessive trait came from the male. In our other case, we have the same mathematics. We have 1/2, 1/2, 1/4, as you can see in the Punnett square. But in this case, the dominant trait came from the sperm, and the recessive trait came from the ova. And then we have our other case, which is still 1/2 times 1/2, or 1/4, in which we had the 2 recessive traits getting together. So this is what we call the law of multiplication, the 1/2 times 1/2, the 2 probabilities multiplied times each other. And then what we see here in the 2 heterozygotes is that if there are 2 different ways to produce what, from our point of view, is the same, is really the same genotype, a heterozygous genotype, then we can simply add those two together, the 1/4 and the 1/4. So we can say that the heterozygotes, in fact, are going to be 1/2. And if we want to go to phenotype, we can go 1/4 plus 1/4 plus 1/4. Those three phenotypes, I should say, those three types of phenotypes were produced by three different ways of combining our alleles. And so we combine those together to be 3/4. So here's our rule of multiplication and addition. The chance that two or more independent events will occur simultaneously, that's this 1/2 and 1/2 that we're talking about as the gametes separate independently. The rule of addition, the probability of an event that can occur in two or more different ways is the sum of the separate probabilities of those ways. And I gave you two examples of that. If we're talking about heterozygotes, then we have two different ways we can produce those, depending on where the dominant allele came from. We have two different ways of producing those, and so the 1/4 plus the 1/4 adds up to 1/2 if we're talking about genotype, the heterozygotes. If we're talking about phenotype, then we have actually three different ways that we can produce the phenotype of purple, and that's by having our homozygous purple and our two heterozygotes, 1/4 times 1/4 times 1/4. Oh, sorry, I said times, but I'm not going back to edit that. 1/4 plus 1/4 plus 1/4, and that becomes 3/4. And now we understand our 3-to-1 ratio in terms of phenotype and our 1-to-1 ratio in terms of genotype. This is our Punnett square. You'll be using this a lot to figure out your genetics problems, and we'll come back to this as we conclude this part of the lecture. There are going to be probabilities in which we're going to deal with that in populations when we get a little bit ahead of that into population genetics as well as an individual. So it kind of works both ways. Okay. So Mendel did a dihybrid cross to explore whether the traits he was examining sorted independently. And he had the P color and he had the P smoothness that he combined together. And boy, was he lucky when he did that, because when he performed that, he did in fact figure out exactly what he was aiming for. And I'm going to go through that step by step here as we explain Mendel's other great advancement. Okay, so here's our dihybrid cross. So you can follow through this in your book, but I've kind of chopped this up so we can see it step by step here. So here we have our two different kinds of true breeding individuals. We have one that always breeds yellow seeds that are perfectly round, and one produces green seeds, which is the recessive trait, that are wrinkled, which is the recessive trait. So these are our starting points genotypically, right? And phenotypically for that matter. Okay, so when these guys produce their gametes, well, there's always going to be the Y in that gamete. That's a separate gene, right? The big Y and the little y. It's always going to have the big Y within this yellow round seed right here. And it's always going to have our big R, which is the dominant trait, round. So all of the gametes are going to have these two separate alleles of those two separate genes, which is color and wrinkled or round. And of course, in your own mind, you should think as well as about a few thousand other gametes that are going to be in there, right? Sorry, a few thousand other alleles that are going to be inside that gamete. Sorry, I misspoke. Then we see in the other true breeding individual where all of the gametes are going to have either the Y recessive trait, which is for green, or the small r, which is for wrinkled. Okay, so we're going to combine those two, and we're going to have our F1 generation, which is going to be round and wrinkled, because sorry, I said it wrong, yellow and round, which is going to be obviously our two dominant traits. But however, of course, now we know that they're heterozygous for both of those two characteristics. But of course, because we have this dominance in terms of color and in terms of being round, they all appear the same phenotypically in the F1 generation. But now each one of these is big Y, little y at that one characteristic, right? And big R, little r at that other characteristic. So now the question that Mendel was trying to answer was about this independent versus dependent assortment. And so, do the alleles of that parent generation stay together were they linked together somehow, the Y for the color and the R for the for wrinkled or smooth, were they linked together in the first place? So we're going to see them come out together again in that F2 generation. Would the big Y, big R, yellow and round stay together, and the little y, little r, green and wrinkled stay together? Because that would be dependent assortment. Okay, that's one possibility, right? And that would look something like this. You would expect to see, again, the 3-to-1 ratio with those parental generations coming out again. What's the other possibility? Well, the other possibility would be that, in fact, they're independently assorting. In other words, that means that the two different traits, whether they have a certain color, whether they're wrinkled or not, are separate from each other. So that means, by our probabilities, that means that when these guys separate up into the gametes and the various alleles get sorted out, the way that Dr. Stemke actually described, we would see up here in the sperm that sometimes, 1/4 of the time, we're going to have big Y, big R, yellow and round. 1/4 of the time, on the other side, we're going to have little y, little r coming together, which would be green and wrinkled. But also, we're going to have the other possibilities, right? It could be that if they're separating separately from each other, then we're going to have our yellow characteristic, our yellow trait, be together with the wrinkled 1/4 of the time, and our green characteristic being sorted with the round 1/4 of the time. Those are our only 4 possibilities for combining these, and they would happen by chance, by probability, just like flipping a coin, 1/4 of the time in the sperm for each of those 4, and 1/4 of the time in the ovum for each of those 4. I know it looks super complicated, right? And that's what it looks like in the end. You start combining those together. Obviously, each one of those, 1/4 times 1/4, are going to happen 1/16 of the time. So here we see our larger Punnett square working like this. And we see all of our 1/16 probabilities coming together. But of course, those are all different genotypes, right? Sort of different genotypes. There are at least different ways in which these genotypes can come together. In some cases, the genotypes are actually the same. Let's take a look at some of those cases. Here we see up here in the top right here, we see a big Y, big R. That was a combination that happened because of where exactly the big Y came from. In this case, from the, we go back to that one, from the ovum. If we look over here, we see that the big Y came from the sperm. So that's a different way to get the exact same genotype. And as you know, we could probably add those together to figure out what that means. In this case, both genotypically and phenotypically. Okay, let's take a look at the next one. Well, we'll add it up. The phenotype, round and yellow, well, we can count them, 9/16. You can count them right here if we just go through that. But how'd that come about? Came about from these different combinations of genotypes. I won't go through listing them all, but you can go through this yourself, and you should go through it yourself to check out how this actually happens. And if we add together 1/6 plus 1/8 plus 1/4 plus 1/8, we get 9/16. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 4 different cases, notice, of the heterozygote right here. Now, the phenotype round and green happens actually in 3 of our 16th times. We have our kind of 2 homozygous for the 2 traits coming together, 1/8, and then we have one that's heterozygous for the round or wrinkled, and homozygous for the color, 1/16. Add those together, you got 3/16. Again, you can go through here and find our round and greens, 1, 2, 3, like that. As you might imagine, that's actually going to match up pretty well with wrinkly and yellow, 1, 2, 3. 3 different ways to come at that. Actually, 2 different genotypes, but remember, in this case, there are kind of 2 ways to come at that genotype. Again, we can add those together. And there's only one way to get the phenotype wrinkly and green, and that's by putting our 2 small y's and our 2 small r's together, and that's our 1/16. So that's our dihybrid cross, and this is the way it would be if there was independent assortment. And in fact, when Mendel actually did his count, he did find out that he had. Here's the numbers right here. Again, not precise because there's always a little chance involved, right? It's random, whether they're 1/4, 1/4, 1/4. Never going to be exactly that way. Any more than if you flip a coin, you're always going to get 5 heads and 5 tails. Sometimes you'll get 6 and 4, 7 and 3. Sometimes you'll even get 10 and 0. But it came out in the expected ratio for the hypothesis of independent assortment, which was 9:3:3:1. That's the phenotypic ratio. And we can go underneath that to the genotypic ratio, and that comes out the way that would be expected as well. So that's the law of independent assortment, and this is a graph that probably Dr. Stemke showed you. Each allele pair segregates independently of the other. Now, I'll go ahead and I'm going to say right now, I should have closed this a long time ago, sorry, but I'm going to go ahead right now and I'm going to say, yes, that's sort of true, the law of independent assortment. And luckily, it was true for Mendel. When I go forward into the next lecture that I'm going to give from section 14.3, we'll talk a little bit more about why that's not true. Probably Dr. Stemke got into that a little bit as well. Or I should say, when it's not true. I just want to say right now, in closing, that of this section on 14.1 and 14.2, I want to go ahead and say that there are simpler ways to do one of these big, 16 type Punnett squares. You can actually solve each of them by 2 by 2 Punnett squares. So if you think about it in that nice mathematical way, you can make your job a little easier. Okay, so if you see right here, we have a parent heterozygous for round and a parent heterozygous for yellow. Like we talked about, we crossed our F1 generation, right? Offspring phenotype, round and yellow. This is the question we're asking ourselves. How would that come about? Well, the chance of being round, if we just look at the roundness of it, we have two, sorry, I should be here. We have 2 heterozygotes, I said, right? Now, this could be in our individuals, our F1 generation we talked about. Well, this is our probability just for being round or wrinkled. It's a 1-to-1 possibility genotypically and 3-to-1 phenotypically. The chance of being round, in fact, is 3/4. You can even look down here and see that that's true. 9 of them are round. I say 9. Sorry, 12 of them are round. The chance of being yellow, in fact, just looking at this very simple heterozygote cross right here, is also 3/4. So what's the chance of being round and yellow? Well, the chance is, in fact, 3/4 our chance of being round and 3/4 our chance of being yellow. If we multiply those together, the chance is 9/16. And remember, that's what we just found. In other words, by simply doing the simple cross just for the individual character, whatever it may be, and then doing your multiplying, when you have a complex dihybrid or trihybrid cross, you can figure out what the result is going to be. What if the offspring phenotype was round and green? Perhaps we can figure this out already. Well, the chance of being round, we already know it's 3/4, right? The chance of being green was in fact 1/4. So that means we can multiply those two together in 3/16. And in fact, if we look over here, we find, in fact, the chance of being round and green is, in fact, 3/16. Without going through all this craziness right here, just by doing these simple crosses and then multiplying the results of these, we can find out what our ultimate numbers would be. And you can do that for genotypes. I did it for phenotype right here. You can do it for genotype. Offspring genotype YYRR. Well, look, the chance of being heterozygous YY is 1/2. The chance of being big R is 1/4. 1/2 times 1/4 is in fact 1/8. And in fact, if you went through this, you would find that 1/8 of these possibilities were in fact heterozygous for the color and homozygous for being round. Now, I know it's complicated. I went through that in about 90 seconds, but of course, you're welcome to sit and stare at this for as long as you want if you pause this particular lecture, and you can go on from there. So I am going to leave. I'm going to stop there. I will later, on my next one, put up some questions that you can ask yourself and see if you can figure out these more complex crosses. So we'll start off my next lecture with that, and I'll do 14.3. But for now, so long, and I'll come back with part 2 a little bit later. I think I'll stop sharing first. Oh, there I am. God, so big. Now, the second. I know at least one of you fell asleep, so I'm going to wake you up. All right. See you guys later. Stop recording.